Welcome back to a new video of the MobileChem channel. Today we're going to talk about the intramolecular and intermolecular whole delocalization that rules the reducer character of isolated nucleobases and DNA strands. So, as everybody may know, DNA is a macromolecule that is in charge of the storing of the genetic code of every single living organism in our world. Its structure is composed of building blocks called nucleotides. Each nucleotide is at the same time composed of apentose, which in the case of DNA is specifically a 2-deoxyribose. This structure is linked to a nucleobase in its carbon 1. There are 5 canonical nucleobases that exist naturally in nucleic acids, although only adenine, cytosine, guanine, and thymine are present in DNA. In addition, each pentose is bonded to a phosphate group in its carbon 5. By undergoing subsequent condensation reactions between the phosphate group of one nucleotide and the hydroxyl group of another nucleotide, we obtain a polymer of nucleotides that is commonly known as a strand. It is also well known that the most common structure of DNA is a double strand, and each strand is antiparallel and complementary one another. The term antiparallel refers to the fact that one strand follows the direction 5' to 3', while the other follows the direction 3' to 5'. In order to obtain this double strand structure, nucleobases from opposite strands interact through hydrogen bonds, stabilizing the shape of this biomolecule. The nucleobases are complementary in the sense that adenine interacts with thymine through hydrogen bonds, while guanine interacts with cytosine thanks to the formation of 3 hydrogen bonds. Although the natural function of DNA is the storing of the genetic code, in the last decades DNA has been used to develop several technological applications such as DNA computation, in which people use specific sequences of nucleotides to carry different information. Another application is the use of DNA to synthesize target molecules, which are not straightforward to obtain by conventional methods. Suppose that we have 2 reactants that give rise to the desired molecule. One of the reactants will be linked to a single-stranded DNA and the other one to its complementary strand. When both strands undergo hybridization, their reactants come closer so that they can react to obtain the target molecule. Moreover, we can take advantage of the capability of DNA to conduct current in order to use this macromolecule as a molecular nanowire. And in this line, we can use DNA for electrochemical molecular recognition purposes. As a result, DNA can act as an electrochemical biosensor by performing a redox reaction with an analyte to detect or even quantify the amount of analyte that is in a given sample. Today we are going to focus in this last application, so let me explain briefly the mode of operation of these DNA-based electrochemical devices. In a first stage, there will be a charge transfer between the analyte and the DNA. This means that the process starts thanks to a redox reaction. Here we can see 2 potential energy surfaces associated to the neutral and the cationic ground state of DNA. This macromolecule is most susceptible to get oxidized rather than to be reduced, so it will likely lose an electron. Thus, when DNA loses an electron, it reaches the cationic potential energy surface with a subsequent relaxation of its structure. This implies that a positive charge, or a hole, is generated in its structure. This hole can be delocalized among the full structure of the nucleotides, but it is well known that in solution, the nucleobases are the responsible moiety for holding that positive charge. Afterwards, the positive charge will be transported along the DNA strand until it reaches a metallic surface. This will lead to the generation of some amount of current along the metal that can be monitored in order to detect or quantify the presence of the analyte in the sample. In this video, we are going to focus in these two first stages of the biosensing process. We are going to determine the one-electron oxidation potential for nucleobases when they are free in water and when they are on a DNA environment. We will also study the whole delocalization after oxidation among the atoms of a nucleobase and along several nucleobases of single-stranded DNA and double-stranded DNA models. We will first explain the computational methodology followed in order to perform these tasks. As explained before, one nucleobase will undergo an oxidation reaction. However, conventionally the redox potentials are given in the direction of the reduction, as you can check here. So we will define the one-electron oxidation potential as a reduction potential of an oxidation process. The free energy of this Hall reaction is related to the adiabatic ionization energy after considering some thermal effects. This energetic term can be defined as the energy required to detach an electron from a molecule and let the system relax geometrically in its cationic potential energy surface. Additionally, the free energy of the process is related to the one-electron oxidation potential by this expression, so our task consists basically on determining the free energy of the Hall redox reaction. A simple way to do this would be to compute the free energy of each individual species that is involved in the reaction. This protocol would be a good approximation for small molecules or rigid molecules that present few degrees of freedom because its potential energy surface will show one clear minimum in energy. The geometry associated to that minimum will be representative of the full phase space, since that conformation will be much more stable than the others. Thus, we can determine the properties of the system computing the properties for that geometry. However, our systems are very complex and so they will be their potential energy surfaces. Many local minima will exist and all of them will contribute to some extent to the macromolecular properties of the system. So we will need to take into account all of them to accurately determine any property. As a result, it will be necessary to sample the conformational space in order to take into account the maximum number of these minima possible and compute the target property for all of them. This can be achieved using molecular dynamic simulations in which we let the system to evolve in time to fetch a representative ensemble of geometries of the conformational space. Thus, if we go back to the former expression for computing the free energy of the reaction, we can rewrite it in order to apply that dynamic methodology. The free energy of each individual species will be replaced by the average of the internal energy computed for every single geometry included in the conformational ensemble. So far so good. We will need to overcome other problematic issues inherent to this study, since both the neutral species and the cation are involved in this half reaction. There exists a need to sample both potential energy surfaces. Using classical molecular dynamics, we need to parameterize them separately because each one will show a different behavior. However, conventional force fields are usually parameterized just for the ground state of the neutral version of the molecule. So what about the potential energy surface of the cation? Well, one possible solution is to perform classical molecular dynamic simulations in its ground state and obtain a high number of conformations that represent the phase space. Afterwards, we can perform further hybrid quantum mechanical molecular dynamic simulations for each geometry of the ensemble in both potential energy surfaces, so that we will be able to relax these initial geometries in the corresponding phase space. The QM region will be determined in terms of the region of interest. Although this protocol is in principle valid, we also wanted to test other alternative methodologies that are commonly used in this kind of studies. So one of them is the formulation of the Marcus theory in dynamic protocols. While the previous schemes compute directly the adiabatic ionization energy related to the one-electron oxidation potential, the dynamic Marcus theory protocol computes two different energetic terms in order to obtain the free energy of the half reaction. The first one is the vertical ionization energy, which is defined as the energy that we need to detach an electron from a system without subsequent geometry relaxation in its cationic potential energy surface. While the second one is the vertical attachment energy, which is the energy released when a cation takes an electron and goes back to its neutral state without geometry relaxation. So in principle, the adiabatic ionization energy, and thus the free energy of the process, can be computed as the mean of both energetic terms, the vertical ionization energy and the vertical attachment energy. In this context, we are not computing anymore the average internal energy of each ensemble, but the average vertical ionization energy and vertical attachment energy in their neutral and cationic ensembles respectively. So once the theory behind the study is already explained, we're going to focus in the main results that we have obtained. We're gonna talk first about the free nucleobases in water and their redox properties, and in a second stage we're gonna talk about these redox properties when the nucleobases are in a DNA environment, and also about the charge transfer along the DNA strand. Okay, so let's go with the free nucleobases in water. Here you can see the computational details that we have used in order to perform these simulations and to compute the redox properties of each of these four nucleobases. Here we can see the one-electron oxidation potentials for each of the four studied nucleobases when they are free in water. Our results are in reasonable agreement with the experimental data extracted from the literature, although our calculations slightly underestimate the redox properties of pyrimidine derivatives, which are cytosine and thymine. However, the relative order of the reducer character is correctly described within these molecules. Purines are more prone to oxidation than pyrimidines. Specifically, guanine is the most susceptible nucleobase to oxidation, followed by adenine, cytosine, and thymine. Not only the one-electron oxidation potential was computed, but also the distribution of the whole among the atoms of each nucleobase upon oxidation. Here I will use guanine as a representative example to discuss the common trends seen for all the nucleobases. The color of the dots placed onto each atom of the molecule represent the amount of whole that is stored in it. We can see that a large amount of positive charge is held in the oxygen and nitrogen of the functional groups, in those carbon atoms that are opposite to the functional groups, and in alternated positions of the ring. These observations are in good agreement with the spatial location of the HOMO orbital from which the electron is detached. In order to measure somehow the delocalization of the whole among the nucleobase, we came up with this empirical formula that takes into account the fraction of the positive charge that is stored in each atom to give an overall value of the delocalization, named intramolecular delocalization number. Here we can see the results of the intramolecular delocalization number for each nucleobase. As can be observed, the larger the localization, the larger reducer character, or what it is the same, the lowest one-electron oxidation potential. This can be related to the extension of the pi system of a nucleobase. Purines as guanine or adenine show a larger pi system that can better stabilize the generated positive charge among a larger number of atoms. Thus, this will lead to a larger susceptibility to get oxidized than pyrimidines, whose pi system is smaller. So now that we understand how the one electron oxidation potential varies among the nucleobases, we can put now these moieties into a DNA-like environment. So as a first approximation, we created 4 different models based on homogeneous single-stranded DNA in which only one nucleotide was present for each strand. This will help us to understand how the one electron oxidation potential varies in a DNA-like environment. Then we used binary combinations of nucleotides in order to generate other 6 models to study how these redox properties were affected in heterogeneous single-stranded DNA. And finally, we did exactly the same with double-stranded DNA, building 6 different models that were representative of binary combinations of nucleotides for each strand. So let's go back to the obtained values of the one electron oxidation potential for nucleobases free in water, and now let's compare them with the analogous results for the homogeneous single-stranded DNA. We can see that there is a decrease of the redox potentials, which can be translated into an increase of the reducer character thanks to the DNA-like environment. Thus, the presence of other nucleobases makes that each nucleobase becomes more prone to oxidation than when it is free in solution. Among these 4 cases, thymine shows the greatest increase in the reducer character. In order to find an explanation, we took the concept of intramolecular delocalization number previously discussed and we reformulated it so that we came up with the intermolecular delocalization number, which is a measure of the number of nucleobases in which the whole is delocalized. The difference between both concepts is the partial charges used. In the intramolecular case, the amount of, we employed the amount of whole stored in each atom, and in the intermolecular case, we applied the amount of whole held by each nucleobase. So here we can observe the values for the intermolecular delocalization number for each strand. We can see that the larger delocalization, the larger increase of the reducer character with respect to the case free in water. Moreover, notice that when there is a large delocalization among a nucleobase, there will be a small delocalization along the strand, giving rise to the fact that there is a competitive effect between both types of delocalization. If the whole is well stabilized in a single nucleobase, which means a high intramolecular delocalization number, there will not be any need of delocalize along the strand, and vice versa. Okay, so we know now that the environment is important when tuning the redox properties of nucleobases, and that the stacked nucleobases increase the reducer character of a moiety because there is a share of vacancy. However, how can the sequence affect the redox potential? Well, we modeled 6 heterogeneous single-stranded DNA with binary combinations of nucleotides. For instance, AC stands for a sequence of alternated adenine and cytosine nucleobases. Afterwards, we determine their one electron oxidation potentials, and here we can see a pink bar that represents the redox potential obtained from that heterogeneous single-stranded DNA. Red and blue horizontal lines account for the redox potential of homogeneous single-stranded DNA of adenine and cytosine respectively. As we can see, the resulting redox potential is just a linear combination of both extreme homogeneous cases, and this can be generalized to all the possible cases according to our results. For instance, adenine N1 strand shows an intermediate one electron oxidation potential between homogeneous cases, which are red and green lines. However, if we study the case in which we have a double-stranded DNA, this trend is not fulfilled anymore. Here we can see an example in which we have a model double-stranded DNA composed of a strand of guanine and its complementary composed exclusively of cytosine. Again, straight lines displayed account for the redox potential of the homogeneous single-stranded DNA of cytosine and guanine involved in this example. So here we can see that thanks to the addition of the complementary strand, the one-electron oxidation potential of the double helix becomes much lower than any of the single-stranded cases, increasing considerably the reducer character of the structure. If we go further and we compare this situation in which guanine are just in one strand and cytosine in its complementary, to the case in which guanine and cytosine are intercalated in both strands, we can see clear differences in terms of their redox potential. In the case in which nucleobases with similar one-electron oxidation potential are stacked, so this case, the reducing character is higher than in the other case. This means that the sequence is relevant in the redox potential of the strand. Although this was just an illustrative case, our results confirmed that these conclusions are general for the double-stranded DNA cases. So the reducer character increases with the addition of the complementary strand, and the sequence is an important factor in the tuning of the redox properties of the system. In general, the more similar the redox potential of two adjacent nucleobases, the higher the reducer character, or what it is the same, the lowest the one-electron oxidation potential. In this line, we were also able to determine the charge transfer mechanism of the whole among the different DNA models that we have been studying up to now. In the literature, two different extreme mechanisms are addressed in order to explain this phenomenon. The first one is called tunneling and states that the positive charge is transported highly delocalized among a large number of nucleobases. On the other hand, the Hopin model claims that the whole is transported localized in just one nucleobase through jumps between nucleobases highly susceptible to oxidation, such as guanine or adenine. Let's take the example of the homogeneous single-stranded DNA composed of guanine. This plot accounts for the percentage of whole allocated in each nucleobase of the strand along a molecular dynamics simulation. It can be clearly seen that around 75% of the whole is held in just one nucleobase, giving some evidence that the Hopin model is more suitable to describe this transport. In order to take our hypothesis, we computed the intermolecular delocalization number for the 16 models that we have been studying. Here we can see the value of the measurement for each model, and if the transport mechanism followed a Hopin model, then the intermolecular delocalization number should be strictly 1, but this is not the case. However, tunneling states that the intermolecular delocalization number should be a high number, around 6 or 7. The vast majority of our models addressed a value lower than 2, giving rise to the fact that there is a large localization of the whole compared to what tunneling states. As a result, we would say that the charge transport mechanism is a mixture of both proposed mechanisms with some predominance of Hopin against tunneling. However, the tunneling model cannot be rejected because there is some delocalization of the whole along this strand. So let's try to summarize all that we have discussed in this video in just 2 or 3 sentences. Well, larger pi systems induce larger delocalization of a whole and increase the reducer character of nucleobases free in water. There is a competitive effect between intramolecular and intermolecular delocalization of the whole when the nucleobases are on a DNA environment. And there is this charge transport that is a mixture of the Hopin model with some extent of tunneling contribution. So this was a summary of the last papers that we have published regarding DNA strands, and I just want to finish saying thank you to the MobileChem group, also the collaborators, and the funding. So see you in the next video of the MobileChem channel, and bye.